Welcome to our second video on topic 7, IB biology, nucleic acids. In this video we are looking at transcription and gene expression. Okay, so we're going to start off looking at the process of transcription. And as the understanding there suggested, it is looking at the direction of that transcription, so that 5 to 3 direction. Remembering what that 5 and 3 means. So 5 meaning the phosphate end and the 3 meaning the hydroxide end of your DNA. So which prime end we're looking at. So here we've got just a very simple diagram of some DNA. We can see the RNA polymerase there, and we can see the process of transcription occurring as the RNA polymerase is reading the template strand. Again, here just a different format of that image that we've just looked at. Remembering your base pairing rule, and that on RNA we've got uracil rather than thymine. So the process of transcription is where segments of DNA or genes are written or transcribed into a molecule of our mRNA, as you know from your core units. You know that this occurs in the nucleus of our eukaryotic cells. It uses the enzyme RNA polymerase. This synthesizes a strand of RNA, which we call pre-RNA. DNA we know is double-stranded, mRNA is single-stranded. Only one of those strands on our double strand of DNA is used as the template strand for transcription. In replication, obviously, both strands are read, but in transcription, only that template strand. So we have two different strands. We've got the sense strand that carries the genetic code for a particular protein, and then we've got the antisense strand, which is complementary to the sense strand. Now, which of these two strands is used for transcription? Well, it's the antisense, and that's because the antisense is the copy, it's the complementary of the sense. Now, the reason the antisense is used for transcription is because that then gives you a copy of that sense strand. Okay, so RNA polymerase knows to transcribe the antisense strand due to the presence of certain promoters. These are short sections of DNA that are non-coding DNA, but they're very, very important for this process. The sense strand contains the code for proteins. The antisense strand is transcribed into the mRNA, so it gives you a copy of that sense strand. It is always in the direction from the 5 to the 3 end. It requires energy to create the bond between the nucleotides, so we call this an anabolic condensation reaction. And energy comes from cleaving the phosphate groups off of the nucleoside, off the nucleoside triphosphates, and they then become nucleotides. The end product of transcription is what we call pre-mRNA. Pre-mRNA needs to undergo a process before becoming mRNA, which we know as mature mRNA. Okay, so looking at these post-transcription modifications. Now, an entire region of DNA is transcribed, but not all of this information is needed to produce a protein. We've got these sections called introns, and these are removed, okay? And exons, so these sections called exons, are spliced back together using what we call spliceosomes. So the introns are removed, the exons are spliced back together. We've got a methyl cap is added to the 5 end, and a polytau is added to the 3 end. So methyl cup at the 5 end, polytau at the 3 end. Remembering what that 5 and 3 means. Now the mRNA is considered mature and it's ready for translation. So until these 3 steps occur, it is still what we call pre-mRNA. Once this has occurred, we've then got our mRNA ready to go on for translation. Now, exons can be spliced together in different sequences. Okay, we call this alternative splicing. So what this does is it enables one gene to code for different versions of proteins. So looking at gene expression now and what splicing has got to do with that. So gene expression is the process of transcribing and translating a gene into a functional protein that carries out a function in the cell or organism. Different cells express different genes. All cells have all the codes, okay, but they don't need to express them all. 3 main things affect gene expression: proteins, acetylation and methylation, and the environmental conditions. Okay, so we're going to start off looking at acetylation, and this is where histones bind to DNA and they block access to the RNA polymerase initiation complex. The tails of histone proteins can be acylated, which will prevent the binding between nucleosomes. This will partially unwind the DNA, giving easier access to the DNA. Acetylation of histones generally keeps the genes active. Deacetylation of histones generally then silences the gene. So acetylation keeps the histones active and keeps the gene active, whereas deacetylation silences the gene. Okay, now we're looking at methylation. DNA is methylated when the methyl group is added to cytosine. DNA that is methylated is not usually expressed. Methylation is long-term. Methylation is what causes the inactivation of the second X chromosome in females. S-adenosylmethionine, SAM, plays an important role here. A 3-dimensional plot showing the relationship between S1-MP2 hypermethylation and clinical parameters of patients. So that's what we're looking here, this 3-dimensional plot. S1-MP2 being a locus that has often been found to be hypermethylated in breast tumour patients. Hypermethylation of S1-MP2 is shown as black circles, whereas no methylation is presented as white circles in this sample of patients. So black circles is hypermethylation of that gene. M and U represent methylated and unmethylated alleles respectively. From the graph, it can be deduced that hypermethylation event was associated with younger patients of a more advanced clinical stage. So when you look at your graph, you've got age, methylation status and stage. So it's all younger. Generally it's younger and at a more advanced stage. You've got a few outliers here. So when we look again at these factors affecting gene expression, acetylation and methylation, we've just got these 2 diagrams here that really show pictorially these 2 different processes. So the effect of environment on gene expression. So we're looking at this concept now of epigenetics, and this is the study of heritable changes in organisms brought about by changes in gene expression rather than the modification of the genetic code. So things such as pollutants, toxins, hormones, temperature, etc. can influence genetic expression. Genes plus environment equals your phenotype. In epigenetic changes, the DNA sequence is not actually altered, but most often a cytosine base will become methylated. This is likely to occur when cytosine is followed by guanine in the sequence. This CpG combination is usually in or near the promoter region. When methylated, the gene is not expressed. The CpG is a genetic tab that can be passed on to daughter cells through mitosis. There is growing evidence that these tags can also be passed on to offspring. Thank you for watching this video, and I'll see you in the next one.